Hey everyone, and welcome to today's Explainer. We are taking a really high-energy, deep dive into the evolution of Epigenome-Wide Association Studies, or EWAS. Specifically, we're looking at the high-throughput technologies that are completely transforming how we process biological data at a massive population scale. If you've been wanting to understand how scientists efficiently analyze complex human traits, deep ancestry, and environmental impacts across huge sample sizes, well, you are in exactly the right place. We're going to explore the fascinating intersection of cutting-edge physical arrays and the advanced bioinformatics ecosystems you actually need to make sense of all that data.
Here's our quick roadmap for today. We'll start with a refresher on DNA methylation basics, track the evolution of array technologies, and then introduce the specialized 270K array. After that, we'll compare the 270K with a 935K, explore how the CD Genomics 270K service completely streamlines this workflow, and wrap up with a look at the future of epigenomics.
Okay, let's dive into this. Section 1. The basics of DNA methylation and our epigenetic foundation.
Before we get into all the incredibly sophisticated hardware and bioinformatics of modern arrays, we really need to ground ourselves in the biology, right? So DNA methylation is essentially the epigenetic foundation of the genome. Biochemically speaking, it's the covalent attachment of methyl groups to DNA. In humans, this happens mostly at cytosine residues located within what we call CGP dinucleotides. Think of DNA as your computer's hardware, the stable underlying genetic code. Methylation, then, acts like a highly dynamic software layer. It signals to the cell which gene should be turned on or off, without ever actually changing the DNA sequence itself. For researchers, decoding these patterns is literally like reading the master instruction manual for gene expression. But because this software layer is so dynamic, constantly shifting based on all kinds of inputs, capturing a true picture of how it functions means you have to analyze it across thousands and thousands of samples.
So why does monitoring this software layer matter so much in modern high-throughput research? Well, it serves a bunch of foundational biological functions. It modulates transcriptional activity, and it maintains chromosomal architecture. But if you're doing epidemiological research, the real goldmine is how it influences cellular development, embryogenesis, and aging. Plus, it directly impacts how the genome responds to environmental exposures. We're talking long-term nutrition, stress, lifestyle factors, the whole shebang. By studying these epigenetic alterations, scientists can untangle the complex mechanisms underlying broad human traits and variations across whole populations. When environmental factors interact with the epigenome, they leave physical marks. Mapping those marks across vast populations is exactly how we understand the precise biochemical pathways that drive complex phenotypic differentiation.
Moving right along to Section 2, the evolution of array technologies, tracking Illumina's progression.
Let's do a quick history lesson on how researchers have actually been measuring these tiny epigenetic marks over the years. Because, as the demand for better, more expansive population data grew, the physical arrays had to evolve. Back in 2011, the Infinium 450K array was a massive breakthrough, letting researchers look at 450,000 distinct CpG locations. It became the total workhorse for early studies. Then, by 2015, coverage expanded huge, with the methylation EPIC v1.0 hitting 850,000 sites. 2022 brought the EPIC v2.0, pushing that all the way up to 935,000 sites. But look at what happens in 2024. The Screening Array 48 kit drops, tracking roughly 270,000 sites. Now, you might be thinking, wait, 270K, isn't that a step backward? Absolutely not. Researchers realized that for massive population-scale studies, the sheer density of the 935K array was creating severe computational bottlenecks and jacking up costs. What the field actually needed was targeted efficiency. So the technology boldly pivoted to optimize purely for massive high-throughput screening.
Which brings us right to Section 3. Enter the 270K array, completely optimized for population scale.
This is the newest innovation in the researcher's toolkit, and it's basically the culmination of years of targeted technological advancement designed specifically to solve those data bottlenecks in large epidemiological studies. Check out this specific number: 269,094 highly targeted CpG sites. And these aren't just randomly selected either. About 100,000 of these are brand new CpG sites derived directly from comprehensive whole genome bisulfite sequencing data. By utilizing that WGBS data, this array captures regions highly enriched in known regulatory functions and cell-specific chromosomal states that the older arrays simply missed. It's an incredibly lean, highly curated, and potent selection of probes designed to maximize your biological signal while totally minimizing computational bloat. And this brilliantly illustrates just how curated this tool is. Every single probe has to earn its keep. When we break down the trait-associated loci, you can see this heavy optimization for epidemiological priorities. You've got over 102,000 probes strictly dedicated to development and aging research, over 44,000 targeted right at mapping environmental exposure signatures, plus tens of thousands of probes specially curated for researching complex inflammation pathways, autoimmunity, and deep ancestry. Since about 50% of these associated loci were deciphered from extensive past array data and global scientific literature, researchers aren't wasting immense compute power filtering through genomic noise anymore. You get to start your analysis strictly on the loci already proven to drive complex human traits.
All right, Section 4, comparing the 270K and 935K arrays. How do you choose the right tool?
This is a critical dilemma when you're designing a new study. Which array is actually better for your goals? The trick here is remembering that more isn't always better. It entirely depends on your application. The 935K array provides incredibly deep coverage, making it fantastic for deep, localized cellular investigations or rare phenotypic research. Things like cancer and rare disease research where you need to search every corner of the genome. But look at the sample throughput, 8 samples at a time. The 270K array, on the other hand, processes 48 samples simultaneously. That is a 6-fold increase in sheer physical throughput. Plus, the sample input requirement drops way down from 250 nanograms to just 50 nanograms. That is absolutely vital when you're working with limited biobank resources. So for population genomics and large-scale environmental epidemiology, the 270K is the ultimate tool. But there's a catch. Processing 48 samples at a time at a population scale generates an immense amount of data, which is exactly why having a dedicated service partner becomes structurally crucial.
That perfectly tees up Section 5, the CD Genomics 270K service, your research partner.
So, knowing the physical power of this hardware, how do researchers actually implement it without their bioinformatics teams drowning in terabytes of raw data? Having the 270K array as a resource is amazing, but unlocking its insights requires a serious engine. This is where the CD Genomics Methylation Screening Array 270K service seamlessly steps in to handle these massive studies. They provide a complete end-to-end bioinformatics workflow. Step 1 is the standard analysis, handing you sample annotation and those foundational raw IDAT files. Step 2 moves into differential methylation analysis, a computationally heavy process identifying specific variations and differentially methylated regions or DMRs across massive groups. Step 3 dives into gene ontology terms to categorize the specific biological processes influenced by these changes. And finally, Step 4 maps it all out through advanced pathway analysis, unveiling the exact biochemical mechanisms underlying the variations. The service takes you directly from a raw physical sample to clear, actionable, mechanistic insights.
And when you're running a massive epidemiological study focusing on really subtle environmental variables, data reliability is literally everything. Minor epigenetic shifts can so easily get lost in technical noise if the processing isn't flawless. Well, after stringent internal testing, the sample-to-sample reproducibility of the CD Genomics 270K service exceeds 98%. For researchers analyzing thousands of biobank samples, that metric represents immense peace of mind. It ensures that the minute variations you're detecting across wide populations are genuine biological signals reflecting real phenotypic traits, completely separated from analytical noise.
When you combine Illumina's highly curated hardware with the CD Genomics rapid processing ecosystem, researchers can move from raw cohort samples to published insights faster than ever before. It's an incredible synergy. First off, unparalleled scalability. Leveraging that 48-sample bead chip format, the CD Genomics service can process up to 600,000 samples a year on a single iScan system. That's wild. Second, it's natively multi-omics ready. The array goes beyond just CpG methylation by strategically including nearly 4,000 high minor allele frequency SNSPs, giving you a deeper genetic context right alongside the epigenetic data. Third, it's incredibly cost-effective, drastically reducing both lab processing costs and bioinformatics turnaround times. And finally, you're operating entirely on curated content expertly selected from global databases, guaranteeing your research targets the absolute most relevant epigenetic landscapes.
Finally, Section 6, the future of epigenomics and integrating multi-omics.
As we wrap up this explainer, we definitely have to look ahead. High-throughput platforms like the 270K aren't just an isolated endpoint. They're a massive stepping stone for the future of the biological sciences. The future of research is all about multi-omics integration.
Imagine researching a complex trait related to cellular aging across, say, 50,000 individuals. By layering robust, cost-effective 270K methylation data with next-generation sequencing technologies like transcriptomics and proteomics, you aren't just looking at static epigenetic markers anymore. You are seeing exactly how those specific markers actively alter biological pathways and protein expressions in real time. This synergistic approach lets researchers map out the intricate relationships between gene regulation and complex traits, unlocking a systems-level understanding of biology that was computationally impossible just a few years ago.
With the incredible high-throughput hardware of the 270K array acting as your resource, and the Genomics Methylation Screening Array service serving as the engine handling everything from sample processing to advanced bioinformatics, population-scale epigenetic research has literally never been more accessible or efficient. So the ultimate question for all the researchers tuning in today: are you ready to scale up your next Epigenome-Wide Association study with the unmatched power of the 270K array? Thank you so much for exploring this fascinating technological evolution with me today. Keep asking big questions, keep exploring the data, and I'll catch you in the next explainer.